Did you know that more than 50% of Americans take 2 prescription drugs daily, and that 20% of Americans take at least 5 prescription medications daily? But have you ever wondered where these pills go? They end up in our streams, rivers, lakes, and oceans. In 2002, a study was conducted by Dana Colpin of the USGS. His team found pharmaceuticals in 80% of the 139 streams that were surveyed across 30 states. Following this breakthrough study, the scientific community has dived deeper into this issue regarding the transport and fate of pharmaceuticals in the water, but there are still a lot of unknowns about how they affect stream processes. Some preliminary work has been done by Emma Rossi that suggests commonly used drugs, like caffeine, affect biofilms in the stream. Biofilms are super important to all aquatic ecosystems. In essence, they are communities comprised of algae, fungi, and bacteria that are held together by a slimy goo. If you've ever picked up a rock from the bottom of a stream and saw that it was green and slimy, you probably just saw a biofilm. They are a fundamental component to the food chain in streams and rivers. I digress. One drug that severely affected biofilms from Rossi's study was ciprofloxacin. Ciprofloxacin is an antibiotic used primarily to treat a variety of infections. As you can see, there are many ways in which ciprofloxacin can enter the water. Rossi's study did have 2 main drawbacks. One, the biofilms were grown on sponges that already had pharmaceuticals on them, so the community that grew on them could have already been altered compared to a reference biofilm in that stream. Secondly, they used really high concentrations that we don't see in the environment yet. It's still unclear how biofilms respond to varying ciprofloxacin concentrations or how biofilm response differs based on their location in the stream. Here at the University of Florida, I'm trying to answer those questions. For those of you who don't know me, hi, I'm Morgan Gallagher, a senior at Virginia Tech studying environmental science and French. This summer, I was awarded a SURF fellowship, which enabled me to spend 10 weeks in the Department of Soil and Water Sciences and work with Dr. A.J. Reisinger to better understand biofilm responses to ciprofloxacin in urban streams. We deployed unglazed ceramic tiles at 4 sites along Sweetwater Branch Creek here in Gainesville, Florida. Sweetwater is unique because it receives a lot of its water from the Gainesville Regional Utility Wastewater Treatment Plant, up to 7.5 million gallons a day. So to investigate the effect of the wastewater treatment plant on biofilms, we selected 2 sites above and 2 sites below the wastewater treatment plant. We predicted that the sites below would not be affected by ciprofloxacin at a low concentration, but would show a slight decrease in their function at medium to high levels of ciprofloxacin. On the other hand, the sites above the wastewater treatment plant would be severely impacted by any dose of ciprofloxacin concentration, and we would see a decrease in their function. So, to test these hypotheses, we let our tiles incubate in the stream for 2.5 weeks. After, we took 20 tiles from each site and placed them into their own mason jar with site water and randomly assigned them a ciprofloxacin treatment dose. Back in the lab, we dosed them with 1 milliliter of their assigned ciprofloxacin concentration every other day for 6 days. After those 6 days, we measured how much oxygen they produced when exposed to the sunlight and then how much oxygen they consumed in the dark. And what we found was kind of shocking. To compare the effects of ciprofloxacin at each site, we calculated response ratios, which is a treatment response divided by the control response, and that's what I will be presenting here. All sites showed a decrease in gross primary production, which is essentially photosynthesis, and so you can see on this graph that all of the responses are below the 1 line compared to the control. And this was particularly noticeable at the wastewater treatment plant sites where there was no measurable gross primary production. For ecosystem respiration, one thing to note is that the wastewater treatment plant site showed an increase in response when given ciprofloxacin compared to our other sites, which didn't really seem to be affected. This increase in ecosystem respiration at the wastewater treatment plant site could be from microbes using ciprofloxacin as an energy source, but we would need to run more tests to be sure of this. Also, we didn't expect our sites above the wastewater treatment plant to fare as well as they did relative to their controls. Remember, we hypothesized that the sites above the wastewater treatment plant would show no function. So one possible explanation is that this was the first time being exposed to these toxins, and so they had enough species diversity to still function even if some other species were impaired. The biggest finding by far was that any dose of ciprofloxacin affects the biofilms negatively. Lastly, some take-home points. The first is properly dispose of your medications. Your local pharmacy has a drug bring-back program, so please use it. Secondly, we humans are altering aquatic system processes and functioning by not removing pharmaceuticals, so we need to get on that. And lastly, this research was made possible by the UF Surf Program, Provost Joseph Glover, University College of Agriculture and Life Science, Mike Nazareth, who directed the surf program, my mentor, AJ Reisinger, and my awesome lab tech, Lindsay Kelly. And so I thank all of you guys. This would not be possible without each and every one of you. If you would like to know more about this research, you can send me an email at the address on the screen, or you can tweet me. If you would like updates on this research, get in contact with me with those ways, or follow updates on my website. And with that, I just want to thank everyone that made this surf experience possible. It was awesome.